Good evening, students. Welcome back to Zoology class. We are talking with principles of inheritance and variation. In that, we are going to discuss sex determination in today's class. See, sex determination is a puzzle to most of the geneticists before the knowledge of the invention of the microscope. After the invention of microscope, later certain and Boveri they have identified these chromosomes. Later on, when there is a cytological studies going on, a person called Hankin has observed a body called X body when he is performing experiments in many of the insects. Okay? Insects, when he is studying the cytological studies and when he is studying spermatogenesis in case of insects, cytological studies and studying spermatogenesis in insects, Henke has identified a body called a specialized structure and he named them as X body. He called it as X body. But this Henkin failed to explain the significance of X body. Later on, other scientists they have told that the X body itself is the X chromosome. Okay. Later on, they have told that this X body is nothing but the X chromosome. Okay. This X body itself is X chromosome. Okay. So, who has discovered this X body means Henkin has discovered this X body. When he has discovered this X body means during conducting experiments on spermatogenesis in insects. Spermatogenesis in insects. Then we have identified this X body. Okay. And later on, there are different methods of sex determination. So, sex determination is of, see here, sex determination. It can be chromosomal method or genetic method. Chromosomal method of sex determination, and the second method is either through genic balance theory. The second method is genic balance theory. Third one is haplodiploidy. Third method is haplodiploidy. And the fourth method, it can be environment influenced. Environment influenced sex determination. Environment influenced sex determination method. So sex determination is by four different methods. One is chromosomal method, second one is genic balance theory, third one haplodiploidy, and fourth one environmental methods. There are four methods of sex determination. We shall go with the first one, chromosomal method of determination. But before going to this chromosomal method, see the chromosomes are broadly categorized into two types. The chromosomes which determine the sex of an offspring are known as allosomes, and the chromosomes other than allosomes are known as autosomes or somatic chromosomes. Okay, so in an individual there are two different sets of chromosomes. Okay, what are they? Autosomes and allosomes. Autosomes are there, somatic chromosomes. Autosomes are somatic chromosomes and they have no role in the determination of sex. Okay? Allosomes are sex chromosomes. The chromosomes which determine the sex of are known as allosomes. Okay, and next, this chromosomal method of sex determination involves heterogamity, whether the males are heterogamous or females are heterogamous. So based on this chromosomal method of sex determination, it is again broadly classified into male heterogamity and female heterogamity. The chromosomal method of sex determination is again divided into two types. Chromosomal method is again divided into two types. So one is male heterogamity and the other is female heterogamity. So the first one, male heterogamity, and the second one is female heterogamity. Male heterogamity and female heterogamity. Now the male heterogamity, see, it is again of two types, that is XXXY method. One is XXXY method and the second one is XXXO method. XXXY and XXXO method of sex determination, that is male heterogamity. Okay, and what are the female heterogamity methods are? See, female heterogamity, ZOZZ and ZWZZ. ZWZZ methods. Okay, so see here, sex determination in case of individuals is first identified by doing some experiments on spermatogenesis in insects by Hankin, and he named that body as X body. Okay, and what he has observed that in the spermatogenesis experiment, not all the sperms has received that X body. Only 50% of the sperms are without X body. Okay, he has discovered, and but he could not able to specify what is the significance of that X body. And later on, the other scientists, they have identified that X body itself as X chromosome. Okay, and then followed by what are the methods of sex determination? There are four methods of sex determination, that is chromosomal method, gene balance theory, haprodiploidy and environmental influenced method of sex determination. Okay, now there are two sets of chromosomes in any individual. They are allosomes and autosomes. The chromosomes other than the sex chromosomes are known as autosomes or somatic chromosomes, and the chromosomes which are involved in determining the sex of an individual, they are known as allosomes. Okay. Now the chromosomal method of sex determination is of two types, that is male heterogamity and female heterogamity. Where you will find these four different types of sex determination, we shall see. See the type of sex determination in case of Drosophila and in case of human beings, it is XXXY method. Okay. So first one, XXXY method of sex determination. Sex determination. XXXY. Where you will find this XXXY method? It can be seen in case of Drosophila and even in case of humans. Okay. In this XXXY method, females females are homogametic. Okay? Here, females are homogametic. They will produce the sperms or they will produce ova of only one type. That all the ova they carry only one X chromosome. Always gametes are haploid. They carry 44. See, in case of Drosophila, 2n is equal to 8 chromosomes. 2n is equal to 8 chromosomes. Out of this 8, 6 are allosomes, 6 are autosomes, and 2 are allosomes. Okay. So the type of sex determination in Drosophila, what I told, females are homogametic. They will produce ova. All the ova carry one X chromosome. They carry one X chromosome. That means 6 plus 1 X chromosome. See here, when it is AAXX, these are females and crossed with male which are having AAXY. The karyotype of this female is AAX, which are these AA are autosomes and XX are allosomes. AAXY is seen in case of males, these are autosomes and XY are males. Okay? And here, all the ova they produce, they contain only AX. Whereas they produce two different types of sperms. 50% of the sperms carry the X chromosome and remaining 50% of the sperms carry Y chromosome. 50% of them carry Y chromosome. Now the offspring will be determined by the fertilizing sperm. If the ovum is fertilized with the sperm carrying X chromosome, then the sex of the offspring is female. If the same ovum fertilized with the sperm carrying Y chromosome AAXY, then the sex of the offspring is male. Y chromosome. Okay. So in case of male heterogamety, the sex of the offspring is determined by the fertilizing sperm. And always sex of the offspring will be determined at the time of fertilization. Okay, so 2 points you need to remember here. Sex of offspring is determined at the time of fertilization.